Good day, this is the Professor, CBMD PhD, and today's Sunday, August 20th, 2023. It is 2:36 and 39 seconds, a.m. Eastern Standard Time, and today's research, proteins and cancer. So let's hop to it here. As scientists reveal how proteins drive growth of multiple cancer types, understanding the molecular basis of cancer may lead to new therapies. Date August 14th, 2023. Source: Washington University. As scientists have completed a deep analysis of the proteins driving cancer across multiple tumor types, information that can't be assessed by genome sequencing alone, understanding how proteins operate in cancer cells raises the prospect of new therapies. As through the past work sequencing the genomes of cancer cells, they've identified almost 300 genes driving cancer. Now, they are studying the details of the machinery these cancer genes set in motion, the proteins and their regulatory networks that actually do the work of causing uncontrolled cell division. They are hopeful this analysis will serve as an important resource for cancer researchers seeking to develop new treatments for many tumor types. The scientific investigators and microbiologists analyzed about 10,000 proteins involved in 10 different types of cancer. They've emphasized the importance of the sheer volume of data in this type of analysis. Many of these important cancer-driving proteins are rare in any single cancer and could not have been identified had the tumor types been studied individually. The analysis included 2 different types of lung cancer, as well as colorectal, ovarian, kidney, head and neck, uterine, pancreatic, breast, and brain cancers. Many of these proteins driving cancer are found in multiple tumor types, but at low frequency. When we analyze many cancer types together, we increase the power we have to detect important proteins that are causing the cancer to grow and spread. A combined analysis also allows them to pinpoint the major common mechanisms driving cancers across types. And beyond the function of individual proteins, such data also allow the researchers to understand how proteins interact with one another to fuel cancer growth. If the levels of 2 proteins correlate with one another, for example, one is present at high levels and the other always is as well, this can indicate that the 2 proteins act as partners. Disrupting the interaction may be a promising way to block tumor growth. Researchers also reveal different ways proteins can be chemically altered to change their function. The researchers documented how such chemical changes, processes called acetylation and phosphorylation, can alter DNA repair, as change immune responses and modify how DNA is folded and packaged, among other important molecular changes that can play roles in driving cancer. But research also shed light on the effectiveness of immunotherapies. Immunotherapies, such as checkpoint inhibitors, often work best in cancers with a lot of mutations, but even then, they don't work for all patients. The researchers found that high numbers of mutations don't always result in an abundance of abnormal proteins, which is what the immune system targets when attacking a tumor. As for some cancers, even with mutations having the potential to generate tumor antigens, if there is no abnormal protein expressed, or very little, such mutations may not be targetable for treatment. This could be an explanation for why some patients don't respond to immunotherapy, even when it seems like they should. As such, the proteomics investigation covering the expression profiles of tumor antigens are particularly useful for designing new immunotherapy targeting selected mutations. In another study, the team identified patterns of DNA methylation and other chemical alteration that can influence the way genes are expressed. And such patterns can be key cancer drivers, and one important finding, the team identified a molecular switch that suppresses the immune system in certain tumor types. In general, the thorough proteomic and chemical modification analysis across many cancer types combine with a long-standing knowledge of cancer genomics, provides another layer of information that they hope can help answer many ongoing questions about how cancer grows and manages to evade many of the best treatments. And this is a professor of microbiology, bioinformatics, pathophysiology, and leader of research. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.